So basically, I'm going to give you a very short introduction, and because a lot of what I was going to cover was very nicely covered on Wednesday by George Bloom, my collaborator, who has been very much involved in the second part of the talk, which will be about our efforts to try to block cell death neuronal cell death associated with Alzheimer's disease, and then and that's that's completely unpublished. The third topic will be a very brief discussion of previously published work, recently published work that looks at neuronal death associated with another injury, ionizing radiation, and then finally a conclusion. So as George pointed out, we know that Alzheimer's disease is a really serious disorder in need of new therapies. Also, as he pointed out, it's basically axiomatic, it's dogma that neurons are post-mitotic, that they don't divide. However, we know that, in fact, we know that if you put in an oncogene or you try to suppress a tumor suppressor gene in a neuron, they die. However, we also recognize that there are places in the brain where there's neurogenesis. And finally, when we look at brains of patients who have Alzheimer's, we find, in addition to the cell death, some fairly substantial evidence for cell cycle reentry. So the hypothesis that George's lab and our lab have been trying to address or test is whether or not there might be small molecules, I'll call them perturbogens, which might be useful to prevent cell cycle reentry and therefore be possibly useful for the treatment of this disease and other diseases. And a point I want to leave you with is sort of an optimistic ending here. I think if we're going to study precision neuroscience or precision any medicine, we obviously need precise diagnostic tools, but we also need precise therapeutic tools. And we are living in a land of opportunity. Right now, you can go on the Zinc database and you can look up 35 million, let me say that again, 35 million compounds that you can purchase. And I would submit to you that within those 35 million compounds, there are precise compounds that are useful either as tools or potentially as the beginning reagents for therapeutics. So we know a lot about cell cycle in mammalian cells. In fact, in 2001, 3 individuals, Hunt, Leland Hartwell, and Paul Nurse won the Nobel Prize for very carefully characterizing, along with many other people, the processes associated with mammalian cell cycle. And this is now high school. My daughter's high school textbook has figures that are like this figure here. I just want to point out a couple of key points. It's pretty easy for us to see proliferation, cytokinesis during mitosis, cell split. You can observe that microscopically. You can monitor DNA synthesis pretty easily by putting in tritiated thymidine, bromodeoxyuridine, or looking for the expression of PCNA. And then there are 2 gap phases in the mammalian cell cycle, first gap and a second gap. And we know, and as you know, we believe that many cells, including neurons, exit the cell cycle somewhere close to a century that looks at the integrity of the genome as well as the integrity of some other macromolecules. These are the so-called checkpoints, the G1 checkpoint or the G2 checkpoint. And another thing that I said as a graduate student, I was trying to force tumor cells that were from the neural crest to differentiate. It's really interesting, and in preparation for this particular meeting, I tried to see what do we know about why do neurons terminally differentiate? I mean, what is the basic fundamental process there, and why do they always stay differentiated? Now, there's some evidence that the Notch pathway is important, some microRNA, but fundamentally, in the decades since I have been studying that, not a lot of progress in terms of the molecular activities has been revealed. In contrast, we know a lot about what are the processes that allow this process to go forward. This is highly simplified diagram. Key players include kinases that are the so-called cyclin-dependent kinases like CDK4, 6, CDK2, and which generally have steady state levels that are changed very much during the cell cycle. What does change are their cognate binding partners, the cyclins, cyclin D1, cyclin E, cyclin B, and these allow for the progression past these checkpoints and through these various phases of the cell cycle. And there are a number of other kinases, such as the aurora kinases, Wee kinase, and phosphatases like CDC25, as well as proteins such as checkpoint inhibitors, CHEK1, CHEK2, that help regulate this process. As George mentioned on Wednesday, there's been almost a decade of literature now that has provided, I think, quite substantial evidence that A-beta 1 to 40, 1 to 42 oligomers, but not monomers and not fibrils, can induce neurons to engage in the cell cycle. And this is just one figure from one paper in which they articulate that. These little red dots indicate uptake of bromodeoxyuridine, and you can see with A-beta you can get a concentration-dependent increase in bromodeoxyuridine stain or in PCNA staining. The other important point that was mentioned by George Bloom on Wednesday was that this process is A-beta dependent. So if you have cells that lack A-beta, you don't see this process. And the other point that was made by his group and others is that this process, the exposure of neurons to A-beta oligomers, activates a cell signaling pathway that includes but isn't limited to activation of AKT, PI3 kinase, the mTOR pathway, and that exposure of cells to inhibitors, for example, inhibitors of that pathway, such as warpmannin or rapamycin, can block that cell cycle progression. So George mentioned on Wednesday that, this model. This is my interpretation of that model. He articulated, I think, very nicely some of the key participants in this process. The big black box in my mind is what happens after the involvement of tau. What is it that encourages the neurons to enter into the cell cycle? What kinds of changes occur? And then how do those changes result in neuronal death? And that's the question that we've been addressing, again, in collaboration with his lab. So I want to show you some experiments that we've been conducting to try to answer that question. These are still a work in progress, but it'll give a little abstract painting of what we're trying to do. We utilize murine primary neurons from wild-type or tau knockout mice that are isolated from the cortex, grown in culture. We then expose them to A-beta oligomers for a 24-hour period, and then we take those two different populations, and we basically look at two things. One, we look at what do those populations spit out into the medium. This we are doing by looking at the supernates using Luminex analyses. I'm not going to talk about that right now, except to mention that, and this is in response to the presentation that we heard by Linda Bergeson, that one of the things that we do see, one interesting change, is a substantial decrement in A-beta, in the expression of or the presence of VEGF in the supernatants of wild-type neurons. And that's, I think, interesting in the context of the importance of VEGF in neuronal survival. But the second thing we've been doing is looking at RNA expression using RT-PCR. We've been using a microarray panel with 84 different genes, and I'd like to just briefly show you some of the results of those studies. So first of all, I think you can see in the heat map up at the top that the profiles in neurons treated with the same concentration of A-beta oligomers for the same period of time, that there's substantial differences in the heat map, just the way in the overall configuration. Second thing I'd like to point out is that if you look at the scale on the wild-type neurons, it's substantially broader than the scale of the tau knockouts. These are just waterfall plots of some of the changes. If we want to drill a little bit deeper and look at what sorts of changes occur, I'd like to call your attention to the right-hand side of the figure, which profiles the changes in the A-beta treated versus the non-treated cells. And this is a 1.5 log deviation from the median. And here I've listed on the right some of the changes in gene expression that we see, as you might expect with any kind of injurious exposure, such as A-beta or ionizing radiation. You see an increase in ATM and ATR. But I think it's really interesting that we also see increases in cyclin-dependent kinase, CDK1, cyclin D1, caspase 3, cyclin A1, And I think equally interesting is a decrease in a protein that is basically blocking these cell cycle dependent kinases, CHEK2. Now, I'd like to contrast that with what we see in the tau knockout neurons. It's a generally muted response. The participants that have increased in the wild type are not seen there. And interestingly, caspase decreases, cyclin D1 decreases, and the cell survival protein BCL2 decreases. So I think those are provocative studies. We've analyzed this data in a greater detail using a variety of computational methods. This is ingenuity pathway analysis. And again, on the left, you'll see some networks that we see. The colored ones are the ones that we probe for. The ones that are not colored are proteins that either interact with or relate to the proteins that we interrogated. I'm not going to go into this in great detail, but I think if you compare, again, the wild type with the neurons, you can see P53, which we heard about a little bit earlier, is upregulated in the wild type neurons, decreased in the knockout neurons. And similarly, there's quite a dramatic difference between these two cells, consistent with the idea that there's a change in gene expression when you expose cells to A-beta oligomers, and that is tau dependent. Okay? So this is just sort of summarizing this first part of work that we haven't published yet. We think that there's a variety of kinases and possibly other proteins that are changed in a tau-dependent manner. I think these are interesting from our standpoint because they're ones that can be treated with a variety of compounds that are available in the pharmacopoeia, including cyclin-dependent kinase inhibitors that are currently under clinical trials for cancer. So this would be a nice example of repurposing drugs that are being studied for other diseases. So now I'd like to just spend a few minutes talking about another model. And this will allow me to show you sort of two orthogonal approaches that we've used using high-throughput screening to try to identify druggable targets and then prosecuting those druggable targets. So I think probably many of you know that another form of stress is ionizing radiation, either by treating a GBM patient, as we heard about earlier from Harry, or exposure to a dirty bomb, or if you happen to be living near Fukushima. There's growing evidence that there's a loss of cognition that's been associated with this exposure to ionizing radiation. We know that ionizing radiation induces tau phosphorylation, and we also know that it causes cell cycle reentry either in culture or in animal models. So again, with the same kind of hypothesis, we speculated that maybe we could identify small molecules that could either prevent or mitigate exposure, the death associated with ionizing radiation. So our overall strategy, and this was started at the University of Pittsburgh, was to take advantage of a library that we had, an siRNA library that was targeting the druggable genome, the human druggable genome. So in this particular library had a little bit over 5,000 pooled siRNAs. The library we have now has about 7,000. So there's 4 different siRNAs in an individual well. We then needed a system that was reproducible, robust, that we could study over a period of time. We utilized a pluripotent stem cell population derived from a teratocarcinoma. This is the NCCI T cells. We basically treated those cells with a dose of ionizing radiation that would cause 50% cell death. That's about 4 gray. And had cells that were exposed to one of those pools of each of the siRNAs. This is a relatively noisy assay, so it was really necessary for us to do it multiple times. And we looked at survival based on a proliferation endpoint, Alamar Blue. In that primary screen, we analyzed the data again because of the noisiness of the data in a number of different ways. We looked at z-scores and also the top 5% of the hits. We looked for those that were overlapping. It's about a little bit over 110 hits. They represent, sort of the general sphere of druggable proteins, some druggable genes, some kinases, some transcription regulators, some GPCRs, a number of enzymes. So then we did a secondary screen in which we used caspase as our endpoint and looked at each of those 4 individual siRNAs, and if 3 out of the 4 prevented cell death or prevented apoptosis, they were examined further. 29 of those came out as positives. So we then looked at those 29 in a variety of ways. One of the ways was to use ingenuity pathway analysis, looked at the networks that came about. One of the top network number 1, is shown here. The darkened circles or triangles or boxes reflect the targets that were actually seen. One of the hits that was quite prominent was the EPA receptor. GRIP3 was another receptor, purinergic receptor. But I'd like to just focus our attention on PI3 kinase, which wasn't actually in our deck of siRNAs, but deduced by this interaction network because that provoked us to consider looking at potential PI3 kinase inhibitors as mitigators of radiation-induced damage. So we used a very dirty inhibitor, widely used in the literature, LY294002, and several others. And I'll just quickly illustrate what this particular slide shows. Our y-axis is Cas-based activity. If you irradiate these cells, you either get a 2 to 3-fold increase in Cas-based expression, activity. Then if you expose those cells 1 hour later to a compound, in this case LY294, there's a concentration-dependent mitigation of that radiation. And you can see that this compound, this is a compound that we'd been working with, a Wart-Mannan analog, also called PX867, that our company was working on. You can see in both cases you can mitigate this in a concentration-dependent manner. We thought, okay, let's go out and mine those 35 million compounds that are out there and just take a handful of PI3 kinase inhibitors that have various levels of promiscuity with respect to their ability to inhibit different isoforms of PI3 kinase, hoping that we might be able to identify the PI3 kinase that was involved. I will tell you without showing you the data that actually all of these can mitigate. So that wasn't really a very good exercise. Many of them have cross targets, but I think it does validate the fact that this particular approach does provide a number of therapeutic agents that are now in clinical trials as possible mitigators of radiation. One of the things we were interested in is, can we take this observation and actually show that you can get an effect in vivo? And I think it's a really neat experiment. You can take a mouse, this happens to be a C57 black mouse, expose it to 9 gray of irradiation, and within 20 days, all of your mice will be dead. If you treat those mice, if you expose those mice to ionizing radiation, wait either 4 hours or 24 hours later, and give a single intraperitoneal dose of either that LY compound I told you about or the other compound that I showed you, actually, you can, even 24 hours later, you can protect 50% of the animals from a lethal dose of radiation. This is not as effective as this compound. So I think this really illustrates that you can utilize this to, in the case of, for example, some sort of toxic exposure to radiation. So this is just sort of summarize what I've said, that these compounds actually seem to be able to block apoptosis. I won't show you the data, but I will tell you in our published work, we've been able to document that protection is due to blocking cell cycle or proliferation of those pluripotent stem cells, and it's due to an ability of those cells now to repair the DNA damage that was caused by that ionizing radiation. So it buys those cells time. Can this actually be looked at in a different way? So this is the second way that we've approached this. Here we've taken 5,000 compounds that we have in a library at the University of Virginia. These are pharmacologically active agents, some FDA approved. This is a list of all of the compounds, and just because of running out of time, I'll just tell you that our primary hit was rapamycin, an mTOR inhibitor. We looked at a bunch of different mTOR inhibitors, and almost all of them were capable of providing protection against the radiation. You can reproduce this by using siRNA with Richter and Raptor. And then finally, we've asked, can we look at neuronal pluripotent stem cells. This was done with Mike McConnell. We see activation after ionizing radiation of the mTOR pathway, and shown here is our ability with mTOR to be able to protect these cells against that ionizing radiation. So I think this provides an opportunity to speculate at least that perhaps we can utilize these therapeutic approaches to prevent neural injury and possibly provide a pharmacological way to mitigate the toxicity of ionizing radiation. So I'd like to just thank the folks in the lab, and I've tried to indicate who they are throughout the talk, also my collaborators at the University of Pittsburgh and the funding agencies, and thank you for sticking around.